Welcome back to On the Edge. I'm Dr. Jonas Moses. This hour we are talking about, this is a special report, and we are talking about public health policy and infectious diseases. We've been speaking in the last two segments with Audrey Serrano, who had a rather serious medical misadventure. Her disease was misdiagnosed, and she was treated on the basis of a misdiagnosis, and it made her quite ill. I want to introduce into the conversation Dr. Andy Maniotis. At the beginning of the end of the last segment, that briefly mentioned you. Dr. Maniotis, Andy, you are the director of the Laboratory for Cell and Developmental Biology at the University of Illinois in Chicago. You are also a special correspondent to On the Edge. As well with me is Dr. Rodney Richards. Thank you very much for joining us, Rodney. I sure appreciate it. And I did want to just give a little bit of background on you. The audience is pretty well known to our audience, Andy. But just to give a little bit of background, Rodney, you received your MS in Organic Chemistry from the University of Denver, and you completed your PhD in Bioorganic Chemistry at the University of Colorado. After you graduated, you worked for a small medical startup biotechnology company, which ultimately became known as Amgen, and I think most of our audience will know Amgen. And during your stay at Amgen in Boulder, Colorado, you pioneered the development of the chemistry and molecular biology that now forms the basis for routine DNA and gene synthesis. Using this technology, you designed and synthesized a novel interferon molecule that was approved by the FDA in 1997 for use as an antiviral drug in the treatment of hepatitis C. You also have numerous other patents issued and regarding diagnostic technologies that you developed while at Amgen, including HIV tests. And I want to stop there for just a second, we'll go a little bit more into your more recent background, to ask you about the kind of test kits that you were working on at Amgen and apparently which are still in use today. Can you talk a little bit about the science?
Yeah, of course. Actually, I started working in the field of diagnostics in 1984 when I was employed with Amgen Incorporated as a collaborative effort with Abbott Laboratories right here in North Chicago.
Okay.
Abbott had perfected the pipeline of taking technology, if you will, and pumping out products. This collaboration had the intent of utilizing Amgen's high-tech, biotech, and fusing that with Abbott's ability to pump these technologies into products. So very early on, I was working in a field called DNA diagnostics, or DNA probes at that time. This was an extension of our ability to create genes from scratch or genetic material from scratch. And these new high-tech probes held the potential to increase dramatically the sensitivity of diagnostic tests. By sensitivity here, I mean detecting smaller and smaller amounts of germs.
Okay.
So that was the brilliance of the technology.
And when you say germ, it's kind of a generic for any infectious agent.
Yeah, that's the nice thing about high-tech, is that once you do it for one germ, you can fairly well apply it to all other germs.
Okay. But was that the case in molecular testing? Or I referred to a conversation we had previously, and I asked you about this, and you mentioned that perhaps when we were looking at the molecular level, we've put our eyes too close to the process, and we're no longer looking at the whole. Yeah, that's correct. Disease.
And I think high tech has its advantages. As I mentioned, higher throughput, lower cost, more sensitive diagnostic tests, but it also has its downsides. And I think it takes us further away from the patient. And I'd like to refer to a quote from a Chinese Buddhist scholar in the year about 600 AD who said the superior physician listens to the voice, the ordinary physician looks at the color, and the inferior physician feels the pulse. So I wondered what he would say about those that look at the molecules of the body. And it's not that you shouldn't look at molecules, it's not that you shouldn't measure the pulse, and it's not that you should not look at the color. But with these high-tech tests, I think they have now formed the foundation of a lot of healthcare providers' treatment strategies, and forgetting that there's the patient out here too. And they need to use their clinical practice to inform their treatment decisions. Many of the tests that we use routinely, such as the antibody tests that were used in your case, they're clearly overused. These tests, in and of themselves, were not intended to form the basis of a diagnosis of infection with HIV, at least according to the Food and Drug Administration and the manufacturers that use them.
In fact, I wonder if I could, can we have the first slide? I just wanted to kind of emphasize this. This is an insert in the test kit, and I wonder if you could read that. Are you able to read that, Rodney?
Yeah, I can. It says, at present, there is no recognized standard for establishing the presence or absence of antibodies to HIV-1 and HIV-2 in human blood.
And this is a current insert, and I might note that
it's like 33 other test kit inserts as well.
There are similar conditions on all of the test kits for this particular infectious disease. My question is, if the manufacturers of these test kits have an issue, in other words, feel compelled to put this kind of caveat on their packaging, why are these kinds of test kits used as the sole determinants for infectious agents?
Well, they should not. Again, the FDA, when they approved the so-called Western blot, which is the confirmatory test for the antibodies that are detected by the screening test, clearly stated that these tests should not be used in asymptomatic populations. In other words, in the absence of symptoms, to inform a clinician's decision of infection, the significance is unknown, as the FDA says. So they should not be used in that manner. However, the various government bodies that we have different functions. For example, the Department of Public Health, the Department of Health and Human Services, the Centers for Disease Control have the mission of public health. Okay, whereas the FDA has the mission of individual health.
I want you to hold that thought. We are going to take a commercial break. When we come back, we'll be talking to Audrey Serrano, Rodney Richards, and Andrew Maniotis. I'm Dr. Jonas Moses. You're watching On the Edge.